Hello everybody. Hope you all are doing well and studying properly. Students, we are starting with new chapter in the unit first, reproduction, and the chapter is sexual reproduction in flowering plant. The previous chapter which we have studied was reproduction in organism, and in that chapter we already have learned about the basic characteristics of asexual and sexual reproductions, their types, and various examples of living organisms following this asexual and sexual reproduction. Now, in this chapter, you will be specifically learning about sexual reproduction in flowering plants. So, this chapter deals with morphology, structure, and process of sexual reproduction in flowering plants, or more particularly in angiosperms. We already have learned that sexual reproduction occurs by formation and fusion of gametes, which then gives rise to a zygote, and that zygote leads to new life. So in order to produce gametes, that is both type of gametes, male as well as female gametes, and to carry out sexual reproduction, the angiosperms consist of a specific reproductive organ, that is flower. Humans and human ancestors have always been using flowers as a symbol for conveying our feelings or gestures towards the others. Even today, flowers are objects of aesthetic, ornamental, social, religious, and cultural values. The flowers are routinely being used in almost every corner of the world for these very purposes. However, for a biologist, flowers are of morphological and embryological importance, and they are the site of sexual reproduction in angiosperms. In 11th class syllabus, you already have learned the structural parts of a typical flower, but still have a quick review of it. In a typical angiosperm flower, there are four holes, or we can say four parts, which are attached to a common part called receptacle or a thalamus. So every part of the angiosperm flower, it is originating from a common part, or they are attached to a common part called receptacle or thalamus. If we go from outside to inside, these floral parts are calyx, which is also called as sepals. Then comes corolla, which is also known as petals. Then comes androecium, which is also known as stamen, and gynoecium, which is also called as carpels or pistil. Here, stamen is a male reproductive part which produces male gametes, that is pollen grains, while the carpel is a female reproductive part which produces female gametes, that is ovule within the ovary. So, these two holes are called essential or reproductive holes, as they serve as most important units of sexual reproduction. While calyx and corolla are referred as non-essential or accessory holes, and they are not participating usually in sexual reproduction process. Students, we already have learned in previous chapter about how a male gamete is transferred to female gamete by the process called pollination and ultimately leads to fertilization. After fertilization, the ovary turns into fruit and formation of seeds takes place from fertilized ovules. Thus, fruits and seeds are the end products of sexual reproduction in angiosperms. And later on, a new plant is produced from these seeds upon germination. Thus, The process of sexual reproduction in angiosperms involves mainly these particular steps. The first step is called as microsporogenesis, which is formation of male reproductive structures called as microspores or pollen grains. Once the microspores and pollen grains are formed, the next stage is followed, that is development of male gametophyte. So second stage is development of male gametophyte. Then third step, it is megasporogenesis. So megasporogenesis is nothing but formation of female reproductive structures called as megaspores. Once the megaspores are formed, they lead to development of female gametophyte, which we usually observe in the form of embryo sac. After the male and female gametes are formed, the next step is pollination. So after attaining the sexual maturity, pollination process will take place. The anthers settle down onto the stigma part and pollen tube will be developed through which the male gametes will travel towards the female gamete. Once the male gamete reach into the embryo sac, they cause fertilization, and this fertilization leads to formation of embryo. By the time the floral parts like sepals and petals, they are getting dehydrated and slowly they will shade away, and the remaining flower parts, they are entering into the formation of fruit, and within the fruit those fertilized ovules will get converted into seeds. So that's how in angiosperms the sexual reproduction is followed. Now let us talk about the structures and events related to pre-fertilization stage. The phytohormones and environmental clues govern the formation of flowers onto the plant shoot. Under the influence of these phytohormones, the shoot apical meristem is transformed into reproductive meristem. This reproductive meristem grows to form inflorescence axis over which floral primordia will develop. The primordia then develops into floral buds and ultimately to the flowers. So within this flower, the androecium and gynoecium differentiate and develop to carry out the sexual reproduction process. Now let us first talk about the male reproductive part of angiosperm flower, that is stamen. A typical stamen is consist of two parts. First, filament and anther. The filament is a long and slender stalk, and its proximal end remains attached to the thalamus structure, or in some cases it also remains attached to the petals of the flower. The anther, however, is a swollen structure present at the other end of the filament. So anther is a fertile or geneseous part. Now let us see how anthers are attached to the filaments, and for this there are various possibilities. The first possibility is that the filament attached to the base of the anther, and this type of attachment is called basifixed type. The second type is termed as adenate, in which the filament connects throughout the length of anther and protrudes out from the apex part. When the filament is attached to the dorsal surface of anther, it is termed as dorsifix type. When the filament is just attached at a point about the middle of the connectives so that the anther can swing on it freely, it is called versatile type of attachment. So these are the four possibilities for attachment of an anther to the filament. Now let us learn in detail about the structural arrangements within the anther. Basically, Anther can be of two types, monothecous. So in monothecous anther, there is only single lobe, and the monothecous type of anthers are mainly found in Malvaceae family. The second type of anther is called dithecous. So in dithecous, there are two lobes, and apart from Malvaceae family, all the other types of angiosperms, almost all the other types of angiosperm flowers are having dithecous anthers. So in this dithecous anthers, two lobes are connected with each other by a sterile tissue called connective. Internally, a dithecous anther is tetralocular. That is, it has four chambers called microsporangia or pollen sacs or the pollen chambers. If we take the transverse section of an anther, we can observe three important parts called anther wall, pollen sacs, and the connective. Now let us learn the arrangement and functions of each of this part. So the first part is anther wall. The anther wall exhibits four layers. If we consider from outside to inside, these layers can be called as epidermis, endothecium, middle layers, and the tapetum. So from outside to inside, the first layer, the outermost layer is epidermis, and the innermost layer is tapetum. Now let us discuss about this each layer and its arrangement and its function. So first layer or the outermost layer, it is epidermis. It is the outermost layer of anther wall. It is single layered and consists of flattened cells. It is protective in function, and that is the only function assigned to this layer. Then the next layer of anther wall is endothecium. Beneath the epidermis is present the second layer of cells called endothecium. It is also a single layer and common for all four pollen chambers. Cells of endothecium layer are radially elongated and they exhibit fibrous thickening of callose. So as the endothecium cells, they are connected to each other. On the walls of these cells, there is a fibrous thickening which is formed due to deposition of callose. However, cells of endothecium present in the shallow groove between two microsporangium are thin-walled and represent the line of dehiscence. So line of dehiscence we can observe from this particular region, anther wall. So as you know that once the anther is getting matured, the anther walls will break up. So this breaking process, it is called as dehiscence process. So the fibrous thickening and hygroscopic nature of these endothelium cells help in the process of dehiscence at the maturity of anther. The next layer of anther wall is middle layer. Middle layer is present inner to the endothecium and made up of parenchymal cells. These parenchymal cells are organized into 1 to 3 layers. The cells of middle layer degenerate when anther reaches to maturity or after the formation of microspores. Due to this degeneration of middle layer cells, the two pollen sacs of each lobe merge to form one chamber in each lobe. So these middle layer cells present in between these two pollen chambers of each lobe, they will get degenerated upon the maturation of anther, and ultimately these two chambers they fuse together forming only one chamber. The next layer of anther wall is tapetum. The innermost layer or the fourth layer of anther wall is a very special layer and it is called as tapetum. Cells of this innermost layer are pyramid shaped, containing dense cytoplasm and more than one diploid nuclei or polyploid nucleus. This multinucleated or polyploid nature is due to the process of endomitosis or endopolyploidy. The tapetum is of two types. The first type is called amoeboid tapetum. Amoeboid tapetal cells or invasive tapetal cells undergo breakdown process and their entire protoplast move towards center part of the pollen chambers. So in center part of the pollen chambers, the protoplast nourishes microspores. The second type of tapetum is called glandular tapetum or secretory tapetum. As the name indicates, these cells are involved in secretion of a very special material called sporopollenin. This sporopollenin is a component of exine layer or the outer layer of pollen grains, and it exhibits fixed type of ornamentation in various types of plant species. This sporopollenin is considered as one of the toughest organic material on earth, and there is no enzyme have been identified in any of the living organism which can degrade the spore pollenin layer. The second type of secretion can be observed as pollen kits. So pollen kit is outermost thick, oily and sticky coating found in certain types of pollen grains. So in those plants where the pollen transfer it is facilitated by insects, so the term for this is entomophilus. So these plants in which the insects are transferring pollens, they are called entomophilus pollen grains. So in all this type of entomophilus pollen grains, the pollen kit is present, which is oily and sticky coating. So because of this sticky nature, the pollen kit helps in attachment of pollen grains with the insect body part, and that's how it travels along with the insect from one flower to another flower. The third secretion from glandular tapetum is compatibility protein. This compatibility protein is very important in pollination process, and it allows a pollen grain which is settling onto the stigma to initiate the pollination process. So all these are the layers of anther wall, and now next part in anther is pollen sac or pollen chamber. The pollen sac or pollen chamber are also referred as microsporangium, as it is the main site where the microsporogenesis or microspore formation process will takes place. In immature anther, a group of compactly arranged sporogenous diploid cells occupies the center of each pollen chamber. Basically, tapetum encloses these sporogenous cells. So as the anther is entering into maturation phase, these sporogenous diploid cells are forming pollen mother cells or microspore mother cells by a step-by-step manner. And these pollen mother cells or microspore mother cells are ultimately going to form the microspores or the pollen grains. The next and the last part of anther, it is connective. Connective is a parenchymatous tissue which is present in between two lobes of diathecous anther. So it means it is connecting these two lobes in diathecous anthers. Not only this, connective is also involved in attachment of the filament and anther. So the filament part of stamen, it is getting attached to the anther part with the help of connective. So all these are the parts which we can observe in transverse section of an anther. So this is it for first lecture. In next lecture, we will study about process of microsporogenesis and the formation of pollen grains or male gametophyte. Till then, goodbye. Students, if you are having any queries or questions related to this topic, you can send those questions to me on my Telegram channel. Once you click onto the links given in description, you will lead to my channel on Telegram. So in there, you can post your queries and questions, and I will try to answer them one by one.